Good evening, everyone. I welcome you all to the lecture series on biomolecules. So today is the lecture second on unit biomolecules. So in lecture first, we study water biomolecules. What are different types of biomolecules? And in that, we get to know that biomolecules are the organic substances that are present inside the living cells. And these biomolecules are non-living, but they have a very high functional role in the living processes. So there are different kinds of biomolecules that we studied, like that I introduced to you in the earlier lecture. For example, amino acids that further form proteins, or lipids, phospholipids, or different kinds of molecules like carbohydrates, or all nucleosides are all come under the category of biomolecules. So in this today's lecture series, we will mainly focus on what are amino acids and what are the proteins as a whole, what are the functional importance of the proteins and how the proteins are formed. So what is the basic structure of proteins? So proteins are the most abundant biological macromolecules. As we in the earlier class, we studied two types of components in biomolecules. One is the biomacromolecule and other one is the biomicromolecules. So amino acids were under the category of macromolecules, but proteins are the but proteins come under the category of macromolecules. So they are the most abundant biological macromolecules that occurs in all the cells and parts of the cells. Proteins also occur in great variety. Thousands of different kinds of proteins are there, ranging in size from relatively a very small peptide to huge polymers, and they vary in their molecular weights as well. So their molecular weight may be in millions, and they may be found in a single cell also. Moreover, proteins exhibit enormous diversity of biological function, and they are the most important final products of the information pathways. So most of the activities that are regulated in our body in the cells are all regulated through the proteins, different kinds of proteins. Proteins are the molecular instruments through which genetic information is expressed. So genetic information that is present in the DNA or RNA, so it gets expressed ultimately in the form of proteins. So DNA, for example, gets transcribed into the RNA by the process which is known as transcription. And this RNA then further leads codes for the proteins by the process which is known as the translation. And these proteins are, therefore, we can say that they are the molecular instruments or the molecular entities through which genetic information is expressed within the cells. Now, what are the building blocks of the proteins? So proteins are made up of smaller units, which are known as the amino acids, and amino acids are the building blocks of the proteins. All the different kinds of proteins which are present universally in any bacteria, for example, or in any living organism are made up of amino acids, and all proteins, whether we isolate a protein from the ancient lines of bacteria or archaebacteria, or we isolate the proteins from the most complex forms of life, we will find that all of these proteins are made up of similar sets of 20 amino acids. So amino acids are more than 20 types, but these 20 types of amino acids are the ones which are present universally in all the proteins. So we can say that proteins are nothing but they are polypeptides. So peptides are the smaller chains of the amino acids. So proteins are polypeptides. When different peptides arranged in a specific manner, and they form bonds in a specific manner to acquire a particular structure that have a functional importance, that have a functional role, they forms the proteins. So proteins are basically polypeptides. They are linear chains of amino acids that are linked by peptide bonds. So this is very important, that which bond is responsible for the formation of proteins. So amino acids in the chain are linked with each other through peptide bonds, and this result in the formation of protein. Now, in the last lecture, we studied one of the important property of amino acids, that these amino acids have two functional groups. The first one is amino group and the second one is the carboxyl group. So the presence of this amino group and this carboxyl groups in the amino acid provides it's the property that allows the polymerization of the amino acids, resulting in the peptide bond formation and ultimately leading to the polymerization of amino acids into the polypeptides or proteins. So the crucial feature of amino acids that allows them to polymerize to form peptides and proteins is the existence of their two identifying chemical groups, that is the amino group NH3 plus and the carboxyl group, that is COO minus. So the amino and carboxyl group can react with each other in head to tail fashion that results in the elimination of a water molecule and thus forming a covalent amide linkage, which in this case of peptides and proteins is typically referred to as peptide bond. So basically what is happening is, if we look into this diagram, what is happening, considering this as a one amino acid and considering this as another amino acid, where R group may be same or R group may be different. So what we can see is that this carboxyl group is reacting with this amino group, and this is resulting in the elimination of water molecule. Therefore, this is a kind of dehydration reaction in which water molecule is being eliminated while formation of a peptide bond. So we can see that this bond which is formed here, amide bond, is in this particular case is known as the peptide bond. So this CONH bond is the bond between the C and this N is this complete orange marked here is the peptide bond, which is formed between the carboxyl group of one amino acid and the amino group of the another amino acid, resulting in the covalent linkage between both the amino acids. And this covalent linkage is known as the peptide bond. So peptide bond formation is a dehydration synthesis reaction, as we see, because what happens when the peptide bond is getting formed, there is an elimination of water molecule. And since there is an elimination of water molecule, this can be known as the dehydration synthesis reaction. The carboxyl group of one amino acid is linked to the amino group of the incoming amino acid, that is to the next amino acid. And in this process, a water molecule is released, thus resulting in the formation of a peptide bond. So this is the structure of the amino acids. This we discussed in the last class also. So just to repeat it again, so to have a better clarity about the structure of amino acids and the proteins. So this is the chiral carbon, in which is also known as the alpha carbon, and all the four valencies of this carbon are fulfilled respectively by first amino group, second carboxyl group, third one is hydrogen, and fourth one is the side chain, which is represented here by R group. This side chain may vary, and based on the presence of like what group is present as a side chain, this amino acid acquires a particular property. So amino acid have a central asymmetric carbon to which an amino group, a carboxyl group, a hydrogen atom, and a side chain groups are attached. And this central carbon is known as the alpha carbon atom. And the properties of the amino acids are based on the side chain group which is present or which is attached to the alpha carbon atom. Also, in the last class, we discussed that the presence of this amino group and the carboxyl group actually gives this amino acid a unique property, which is known as the zwitterine formation property. So in this, this amino group and the carboxyl group, due to the presence of these two groups, amino acid may exist as a zwitterine. Zwitterine is the condition where the net charge is negative, where the net charge is 0. So in that condition, the amino acid is known as a amino acid is known to exist in the zwitterionic form. So this was how the peptide bond formation occur. Also, this is since this is in this the water molecule is getting eliminated. So as I mentioned earlier, that this is known as the dehydration reaction, but it may also be called as a condensation reaction because of the elimination of water molecules. So the peptide bond formation is, we can say, is either a condensation reaction or it's a dehydration synthesis reaction in which the water molecule is eliminated and uh, result into the peptide bond formation. So these are the different side chains that may be present. These are the different amino acids that are 20 amino acids that I talked about. So in all of these 20 amino acids, we can see that it is the pink highlighted R group or the side chain that differs. And based on what are the side chains that are present, what is the group that is present as a side chain, the amino acid has a particular property. For example, in this we can see that the presence of just simply the hydrogen imparts the property of being it as non-polar, while on the other hand, if I look at look at the phenylalanine, in the phenylalanine we can see that there is a presence of an aromatic ring. So this aromatic ring basically imparts the aromaticity to the amino acid, and this phenylalanine comes under the category of aromatic amino acids. Similarly, histidine, arginine, and lysine are the positively charged, have positively charged R groups or positively charged side chains. Therefore, they are positively charged amino acids, or aspartate and glutamate are the negatively charged amino acids. While there are some amino acids which are which have uncharged side groups, but they are polar in nature. Example: serine, threonine, cysteine, asparagine, and glutamine. Similarly, we can see that in case of serine and threonine, there is the presence of OH group in the side chain. Therefore, serine and threonine are also known to contain alcoholic groups. So they also can be called as the alcohol amino acids. So this imparts the polarity to these amino acids, the presence of this OH. And cysteine and methionine are known to contain the SH group. So this is just a simple diagrammatic representation of all the amino acids, all the 20 amino acids that are mainly present in the proteins. So properties of the proteins, as I mentioned, depends on the sequence of amino acids and the configuration of protein molecules. Like what is the sequence of amino acids, and in what fashion these amino acids are arranged, and what are the bonds that are formed so that a particular molecule acquires a particular configuration. All these things imparts a particular property to a protein. So variation in the amino acids depend on the side group, as I mentioned, and as it is clearly visible in this figure, we can see that glycine is the one which is the simplest amino acid because its R group or the side chain group is the simplest, which is simply just the hydrogen. And except glycine, all of these amino acids have a chiral carbon in which all the 4 valencies are fulfilled by different atoms, while in case of glycine there are 2 hydrogen atoms which are attached to the same carbon. So in case of glycine, the alpha carbon or the central carbon in which all the 4 valencies are not fulfilled by the different atoms, therefore it is not a chiral carbon, while in other cases it is a chiral carbon. So glycine is the simplest amino acid. Cysteine and methionine, as I mentioned, are the ones that contain the sulfur. So they are sulfur containing amino acids, as we can see in this diagram. So cysteine is here, which contains sulfur, and methionine also contains sulfur. So therefore, cysteine and methionine are the ones that contains the sulfur. So they are sulfur containing amino acids. Tryptophan is the most complex amino acid, as we can see in the diagram. So tryptophan is the most complex amino acid. Tyrosine helps in the synthesis of melanin pigment, thyroxine hormone, adrenaline, which is epinephrine hormone, noradrenaline, norepinephrine hormone. In proline, in case of proline, if we will see, so proline is a special case again. So in this case, if we will look into the figure of the proline, what we will find is that in case of proline, instead of amino group, that is instead of NH2 group, there is a presence of NH group. So this is the amino group which is present instead of amino group. So this amino acid is also known as the amino acid. So instead of NH2 group, there is a presence of NH group in this. So as we can see that in the other NH2 group is present. So instead of NH2 in this case, NH group is present. So which is an amino group. So therefore, we can also call this amino acid as an amino acid. Tyrosine, tryptophan, and phenylalanine are the aromatic amino acids. Proline, histidine are heterocyclic amino acids because they have heterocyclic rings. So we can see in this figure also that proline and histidine are the heterocyclic because they contain the heterocyclic rings. So they are also known as the heterocyclic amino acids. Serine and threonine are alcoholic amino acids because they have OH group in their structure. So based like the property of amino acid is clearly defined or based on the side chain group which is present in the amino acids. And based on these and based on the sequence of amino acid, like which amino acid is present in the sequence of a protein, the sequence of the amino acid defines the properties of the protein. So ultimately, the structure of the protein, the properties of the properties of the protein depends on the sequence of the protein, amino acids in the protein. Now, amino acids can be classified into two categories. The one is essential amino acids and the other one is non-essential amino acids. So it may be confusing to some, like if you listen it to first time, like essential amino acids may be considered as the ones which are important for us, while we can say that non-essential are the ones that are not important for us. But this is actually not the case. The word essential and non-essential here in terms of amino acids is not based on whether they are important for us or whether they are not important for us. In fact, it is based on the fact that whether they are already synthesized by our body or we have to take them from some external source in the form of our diet. So basically, essential amino acids are the ones that our body cannot synthesize and that we need from some out sources, like from our diet. While the non-essential amino acids are the ones that our body can synthesize and therefore we do not need them from the out sources. And so this is the actual definition of essential and non-essential amino acids, that the amino acids which our body can synthesize are the non-essential amino acids, and as we do not need to take them from some other sources, while the essential amino acids are the ones that our body cannot synthesize and we need them in our food supplements. So this is the list of essential amino acids. Essential amino acids, there are 10 essential, out of 20, 10 are essential amino acids and 10 are non-essential amino acids. So arginine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine are the essential amino acids, while alanine, asparagine, aspartate, cysteine, glutamate, glutamine, glycine, proline, serine, and tyrosine are non-essential amino acids. So it is very important to know that which are the essential amino acids and which are the non-essential amino acids. I'll share the mnemonics to remember the essential and non-essential amino acids with you. So now, what are the functions of proteins in our body? So proteins carry out many functions in living organisms. They carry out the function of transportation. For example, we know hemoglobin. It helps in the transportation of oxygen. So some transport nutrients across the cell membrane. We know like there are some proteins that are present in the cell membrane itself. For example, the integral membrane proteins or the peripheral proteins. Some fight infectious organisms, some are hormones, some are enzymes, etc. So they have a wide variety of function. They play a wide variety of functions. They have a wide variety of functions and they play many important roles in the living organisms. So they may help in the transportation. They may help in fighting the against the infection. They may they may be act as hormones or they may act as enzymes. Now, what are the most abundant proteins in animal world? So collagen is the most abundant protein in the animal world. While if I talk about the plant proteins, so in plant proteins or in the whole biosphere, we can say rubisco, that is ribulose bisphosphate carboxylase oxygenase. So ribulose bisphosphate carboxylase oxygenase, rubisco is the most abundant protein that in the whole biosphere. So if the question demands like which is the most abundant protein in the animal world, so the answer should be collagen. But if it asks which is the most abundant protein, so the our first priority should be rubisco. And only if the rubisco is not there, then the question might be related to animals. So in that case, collagen must be the answer. So there are different proteins. Some of them are mentioned here and with their functions. So the first one is collagen, which I mentioned also that it is the most abundant protein in the animal world. So what is the function of collagen? It is an intercellular ground substance. Next is trypsin. So trypsin acts as an enzyme. Insulin is a hormone. Antibody that fight infectious agents. Receptors, for example, the various receptors which are present for the smell, taste, and which are present for the like ligand receptor reactions. So some of them are sensory reception, some of them have a role in sensory reception. Next is GLUT4 that helps in the transportation of glucose. So it enables glucose transport into the cells. So like as I mentioned that the proteins may help in the transportation of nutrients. They may help in fighting against the infection in the form of antibodies. They may act as hormones, for example, insulin. They may act as enzymes, for example, trypsin. So these are the various functions, wide variety of functions that proteins may carry out. So proteins there is a level of organization in proteins as well. Proteins occur in different forms. So there are different structures of the proteins. So proteins, as I mentioned, are heteropolymers that contain strings of amino acids. By term heteropolymers, I mean that polymers may be it is a polymer. Why we are calling it as a polymer? Because it is made up of different amino acids. So it is made up of multiple units. So that is why it is polymer. Why it is called as heteropolymer? Because it is made up of different units. So different amino acids combine to form a protein. So since different amino acids, for example, arginine may form a link with lysine, lysine then form a link with glutamate. So in this fashion, different amino acids may be arranged. So since different amino acids are arranged, therefore it is a heteropolymer. So proteins are heteropolymers that contains the strings of amino acids. And the structure of the protein is described at 4 levels as I showed in this, as it is shown in this diagram. So the first level is the primary level, second is the secondary, third one is the tertiary, and the fourth one is the quaternary. So with each level, the complexity increases. So in the primary structure, it is simply the amino acid sequence, and simply the amino acid sequence of a protein defines the primary structure of the protein. And as we can see that there is a N group, like amino group, N-terminal amino acid, and there is a carboxyl group at the end, which defines the C-terminal. So the last amino acid is called the C-terminal amino acid, and the first amino acid is called as the N-terminal amino acid. So the protein chain is always from N to C. So the first amino acid is the N-terminal amino acid, in which the N-terminal is free, while the last amino acid is the C-terminal amino acid, in which the carboxyl terminal is free. So this is how the protein is read from N to C. And the primary structure is simply defined by the amino acid sequence which is present in the protein. If I talk about the secondary structure, so in secondary structures, so in primary structure, only the bond which is present is the peptide bond, which is present in between the different amino acids. But when there is a bond formation between the amino acids itself, between the different amino acids, except the peptide bond formation, when there are other bond formation, it may lead to the formation of more complex levels. So the secondary level is the secondary structure formation. And in this case, the hydrogen bonding interaction between the adjacent amino acid residues of the polypeptide chain can allow the rearrangement or the arrangement of the protein into the characteristic helical or the beta plated sheet structures, as we can see in this. So the adjacent amino acids may form hydrogen bond with each other, and these residues may either like this hydrogen bond formation may either lead to the alpha helical sheet formation or alpha helical formation or the beta pleated sheet formation. So these segments constitute structural conformities, so-called regular structures that extend along one dimension like the coils of a spring. Such architectural features of a protein are designated as secondary structure, and in proteins only right-handed alpha helices are observed. So secondary structures are just one of the higher levels of a structure that represent the 3D arrangement of the polypeptide in space. So in this, if I repeat it again, so secondary structure includes the alpha helix and the beta sheet due to the hydrogen bond formation in between the adjacent amino acid residues. The third level is the tertiary structure formation. So when the polypeptide chains of the protein molecules bend and fold in order to assume a more compact three-dimensional shape, it is called as a tertiary structure. And tertiary structure is necessary for the biological activities of a protein. So a protein is biologically active when it is present in its tertiary structure form. So in this case, when when there is a further bending and folding, and there is further bond formation in between the different amino acids, it leads to a more complex and a more compact three-dimensional structure, which is known as the tertiary level of structure in proteins. And the most important point is tertiary structure is necessary for the many biological activities of protein. And it is by the virtue of this tertiary structure that proteins adopt a globular shape. So we know about globular proteins. We will get to know about fibrous proteins. So most of the proteins which adopt a globular shape by the virtue of their tertiary structure. And what is the advantage of acquiring this globular shape? So a globular conformation gives the lowest surface to volume ratio, minimizing the interaction of proteins with their surrounding environment. So basically, this globular shape is attained by the virtue of the tertiary structure formation. And why it is important? It is important because when it acquires a globular conformation, the surface to volume ratio decreases, and this minimizes the interaction of the protein with the surrounding environments. Now, what are the quaternary structures, which is the ultimate last level of protein structure? So many proteins consist of two or more interacting polypeptide chains. So when more than one polypeptide chain is present and these polypeptide chains interact with each other, so these polypeptide chains in their tertiary structure interact with each other. So this is leads to the formation of a quaternary structure formation of protein. And these different polypeptide chains may be called as the subunit of the proteins. So this subunit organization constitutes another level in the hierarchy of protein structure defined as the protein's quaternary structure. So these are the four different levels of the structure of the protein. The first one is the primary structure, in which there is just simply the chain of the amino acids leading to the formation of a polypeptide. Then there is a secondary structure level in which there is a formation of alpha helix or beta sheets by the virtue of the hydrogen bond formation between the adjacent amino acids. Then there is the third level, which is known as the tertiary structural level, in which these beta pleated sheets or these alpha helical sheets interact with each other through various other bonds, like for example in disulfide bond as represented in this diagram. So they interact with each other and resulting in the tertiary structure of the protein. And in many proteins, this tertiary structural level is the biologically functional state of the protein. in some cases, there may be a quaternary structure of the protein in which different polypeptides, for example, different polypeptides in their tertiary structure, for example, this A polypeptide is interacting with polypeptide B and resulting in the quaternary structure of the protein. So these are the different structural levels of the protein. So the primary structure of a protein is determined, as I mentioned, that the primary structure, that is just the polypeptide chain structure, is determined by the covalently linked amino acid residues in the polypeptide backbone. So only the peptide bond is present in, if I talk about the primary structure of the protein. But in other higher orders, like in secondary structure or in tertiary or quaternary structures, these are determined principally by the non-covalent forces such as hydrogen bonds, as we see in the case of alpha helix or beta pleated sheets, or they may be governed by ionic bonds, van der Waals interactions, or hydrophobic interactions. So this is the cartoon that is showing a secondary structure in the form of alpha helix and a tertiary structure of the protein. So here, how can we identify it is a tertiary structure? We can see that there is only one N terminal and one C terminal, and the protein starts from the N terminal to C terminal. So there is only one polypeptide chain that has acquired a tertiary structure in this case. So this is a tertiary structure. If in case a similar another polypeptide chain is present and there is an interaction between those two polypeptide chains, then the structure would be known as the quaternary structure. So what are the different bonds, as I mentioned, that are present in the different orders or different hierarchy of proteins? So first one is the peptide bond, which is the strongest bond in the protein. It is present in the primary structure itself. Second is the hydrogen bond that is present in the secondary structures. Third is the disulfide bond. These bonds are formed between the SH group of the amino acid cysteine. These bonds are the second strongest bond and stabilize the tertiary structure of the protein. So we can see that in this case, if there are two cysteines that are present, let's say two cysteines are present far from each other in a polypeptide chain, and these two sulfur molecules of these two cysteines form a disulfide bonds. So these disulfide bonds help in stabilization of the tertiary structure of the protein, and they are the second strongest bond after the peptide bond in case of proteins. Then there might be a hydrophobic interaction between amino acids which have a hydrophobic side chains, for example, aromatic amino acids. There may be an ionic bond formation. Formation of ionic bond occurs between two opposite ends of a protein molecule due to the electrostatic attraction. Or there may be some van der Waals attractions as well. So these are the different interactions or the different bonds that may be formed in different levels of the hierarchy of the protein structure. So these are peptide bonds, hydrogen bond, disulfide, hydrophobic bonds, ionic bonds, etc. So now the proteins can be classified. So now we get to know what is the structure of protein, how the proteins are made, like they are made up of amino acids, so they are polypeptides. What is the different structural hierarchy of the protein, which is the functional unit of the protein, and which is the, like tertiary structure is the most common biological functional structure of any protein, of most of the proteins. And we get to know about different amino acids that are present in the protein, how this peptide bond is formed between the two amino acids, and what are the functions of various proteins. So this slide explains the classification of proteins. The proteins can be classified either as simple, conjugated, or derived. So as the name suggests, simple is just the complete that is made up only of amino acids, while the conjugated, as the name suggests, is made up of, these are complex proteins that are combined with characteristics of non-amino acid substances, which are called as the prosthetic groups, and a amino part. That is, for example, nucleoproteins. So it consists of some nucleic acids and proteins. So it is made up of combination of a protein part and a non-protein part. And the third one is a derived protein. So this derived proteins are the ones that are basically they can further be classified as primary and secondary. So primary are the ones that are formed by the denaturation process. Secondary are the ones that are formed by the process of digestion by the help of various enzymes. So example, simple proteins can further be divided into globular proteins or scleroproteins. Scleroproteins are also known as fibrous proteins. So globular proteins examples include albumin, globulin, prolamins, gluteins, histones, protamins, and globins. While scleroproteins examples include collagens, elastins, and keratins. So collagen, as I mentioned, is the most abundant protein in animal body, and the conjugated proteins include the nucleoproteins, chromoprotein, glycoproteins, phosphoproteins, lipoproteins, metalloproteins. So they include a protein part that is made up of amino acid and a non-protein part which is a non-amino acid part that is also known as the prosthetic group. So as I mentioned that in case of nucleoproteins, it is a combination of proteins and nucleic acids. Chromoproteins, for example, is a combination of proteins and some colored pigments. Glycoproteins are examples are combination of proteins and carbohydrates. Phosphoproteins are phosphoproteins are a combination of proteins and phosphate group. Lipoproteins are a combination of proteins and lipids. And metalloproteins are the combination of proteins and metal ions. So in conjugated proteins, basically a protein part is present and a non-amino acid derived a non-amino acid group is present. Now, the derived proteins are simply the primary and secondary. So, as I mentioned, primary are the ones which are formed by denaturation. Examples may include coagulated proteins, proteins, and metaproteins. Secondary derived proteins are the ones that are formed by the digestion. For example, the protein may get digested with the help of the enzyme proteases, resulting in peptones that further leads to polypeptide formation, that further leads to peptide formation and ultimately resulting into the amino acids. So these are secondary proteins, secondary derived proteins that are formed as a process of digestion of the proteins. So this, sorry. So this was the classification of the proteins. Now what do I mean by the protein denaturation? So protein remains in a particular state, in a particular structure, in a particular environmental condition. Changes in those conditions may result in the changes in the structure of the protein. So changes in the pH, salt concentration or the salt condition, heavy metals, temperature, pressure, etc. may also cause the precipitation or denaturation of the proteins. And because of these changes, what happens is there is a change in the secondary and tertiary configuration of the proteins, and these secondary or tertiary configuration may get destroyed, and many times the tertiary structured globular proteins become converted to secondary configuration fibrous protein. Such alteration in the physical state of the proteins is called as the denaturation. Now this denaturation may be temporary or permanent. So if the changes in the medium of the protein is mild and for a short period, then the denaturation of the protein is temporary. However, if the change in the medium is so strong and prolonged, then the denaturation is permanent and the protein becomes coagulated. For example, we have heard of the white or albumin of the egg. So this is a proteinaceous part. So this is a soluble globular protein. But what happens is when we heat it, due to the excessive heating, it permanently coagulates into fibrous insoluble form. So this is a permanent denaturation, and it is clear that strong alterations result in the denaturation of proteins and they lose their biological properties and significance. So this is the reason that cells of organisms are unable to bear strong changes, as these changes may result in the changes in the structure, secondary or tertiary structure of the protein, which may sometimes be irreversible. And therefore, if these changes become irreversible, protein will become non-functional and the biological properties of the protein might change, and it may result ultimately into the death of the cells. So it is therefore very important that the cells of the organisms are that is why maintains a particular pH, particular salt concentration, and works at a particular temperature. So this is about the proteins. Now, just a quick revision. So what are the different types of proteins? What are the function of proteins? Proteins are made up of amino acids, mostly 20 amino, all the proteins that are present, either we talk about from the ancient bacteria to the current life forms, complex forms of life, all of them are constructed from the same set of 20 amino acids. The property of amino acids is based on the R group which is present or the side group which is present attached to the alpha carbon atom. Also, during the formation of the polypeptide chain or the peptide chain during the formation of during the formation of polypeptide chain, there is a peptide bond formation, and this peptide bond formation results in the removal of a water molecule, and therefore this reaction is also known as the dehydration reaction. Then we studied different properties of the amino acids based on the different R groups that are present with the different amino acids that are attached to the alpha carbon atom. Then we get to know what are the essential amino acids and what are non-essential amino acids and why they are called so. And we study about functions of various proteins. What are the different hierarchy of the structural hierarchy in the proteins? So sorry. So proteins can be proteins can acquire primary structure, then they have a secondary structure, tertiary structure, and a quaternary structure. Then we get to know what are the different bonds that are present in a primary structure and in secondary, tertiary, or quaternary structures. What are different types of proteins? How can we classify proteins? So we classified proteins in three different types: simple, conjugated, and derived. Simple proteins were further classified into globular proteins and scleroproteins, which are also known as fibrous proteins. The second form was conjugated proteins. Third one was derived proteins that were further classified into primary and secondary. Then we studied what is denaturation. Denaturation can be temporary or permanent. So we studied an example of white or albumin of egg, which is a soluble globular protein, but upon heating, it coagulates permanently into fibrous insoluble form. Now, there are some questions that are based on today's discussion. So, first question is the simplest amino acid having only a single hydrogen for an R group is. So, options are valine, option 2 glycine, option 3 histidine, option 4 alanine. So, it is very clear that we studied that the simplest amino acid is the glycine, in which the R group is hydrogen atom itself. So the answer to this question is option number 2, glycine. Question number 2 is peptide bond formation is an example of which kind of reaction? So options are option 1, condensation, option 2, hydrolysis, option 3, deamination, option 4, transformation. So we know that during the formation of peptide bond between the carboxyl group of the first amino acid and the amino group of the upcoming amino acid, there is a release of a water molecule. During the release of the water molecule, results in the peptide bond formation and the linkage of these two amino acids. So, since there is a release of water molecule, the reaction was known as the dehydration synthesis reaction, or we can also call it as a condensation reaction. So, therefore, option number 1, which states that peptide bond formation is an example of the condensation reaction, is correct. Question number 3, the characteristic helical or pleated segments formed through hydrogen bonding interactions between adjacent amino acid residues in the polypeptide chain of protein is seen in. So it is based on this question is based on the structural hierarchy of the proteins. So in this question, what we are asked is that the characteristic helical or the beta pleated segments that are formed by the through the hydrogen bonding interactions between the adjacent amino acid residues comes under which structural hierarchy? So the options are primary structure, tertiary structure, secondary structure, and quaternary structure. So we know that in the primary structure, it is simply the chain of the amino acids and includes only the peptide bonds. In tertiary structure, it includes alpha helix and beta sheets. Also, it includes the disulfide bond formation that in which these get band and acquire a particular configuration. In the secondary structure, there is a formation of alpha helix and beta pleated sheets by the hydrogen bonding between the adjacent amino acids. So the characteristic helical or pleated segments formed through the hydrogen bonding interactions between adjacent amino acid residues in the polypeptide chain of the protein is seen in option number 3, secondary structure. This is again based on the structural like level of protein organization. So we have to tell which level of protein organization is in the given figure. So the first one shows just the chain of amino acids. Second one here shows the alpha helix formation. Third one is a polypeptide chain which is arranged, which has acquired a particular configuration. And fourth is the assembled subunits. So we know that the amino acid residue, the chain of the amino acid residue is represented by the primary structure of the protein. Alpha helix represents the secondary structure of the protein. A particular 3D configuration of polypeptide chain is represented in the tertiary structure of the protein, and different subunits assemble to give the quaternary structure of the protein. So the different levels of organization, protein organization, which are shown in the given figure are primary structure by this amino acid residues, alpha helix represents secondary structure, polypeptide chain represents the tertiary structure, and assembled subunits represents the quaternary structure. Next question, question number 5 is unfolding of a protein can be termed as. So we know that the options are, firstly we should know read the options. Option number 1 is renaturation, option number 2 is denaturation, option number 3 is oxidation, option number 4 is reduction. So renaturation is the reverse of denaturation, in which when I talked about temporary denaturation. So if I remove the conditions under which the denaturation has happened, only the mild conditions in which there is a temporary denaturation, and if I revert back the conditions to original and protein acquires its original configuration, then the process is known as the renaturation. So unfolding of the protein can be termed as a denaturation, option number B, in which if we change the pH, salt concentration, or the metal ion concentrations, or the temperature around the protein may change its conformation and it may unfold, or the process is known as the denaturation. Next question is primary structure of the protein is due to the presence of option number 1 hydrogen bonds, option number 2 peptide bonds, option number 3 disulfide linkage, option number 4 ionic bonds. So we know what is the, firstly we should know what is the primary structure of a protein. So we know that primary structure of a protein is simply defined by the sequence of the amino acids. So in primary structure, how we represent a primary structure? We just represent the primary structure by different amino acids that are linked to each other through the peptide bonds. So the correct answer to the question primary structure of the protein is due to the presence of option number 2 peptide bonds. Next question is the structure of the protein which gives a three-dimensional view is primary structure, alpha helix, beta pleated sheet, or tertiary structures. So we know again primary structure is the one in which is simply defined by the sequence of the amino acids linked to the peptide bonds. Alpha helix is the secondary structure formation, is a secondary structural level of organization in the protein in which the adjacent amino acids may form hydrogen bonds with each other and form a staircase-like structure which is known as the alpha helix. In the beta pleated sheets, different sheet-like structures are formed due to the hydrogen bond interactions between the amino acids, and it is also a secondary structural representation. And while option number 4 tertiary structure is the one in which there is a bending in the polypeptide chain, there is an arrangement of the polypeptide chain in a particular conformation, giving it a three-dimensional view, imparting it the functional properties, and so therefore the structure of the protein which gives a three-dimensional view is the tertiary structure, and most of the proteins are functional in its tertiary structure form. Next question is which amino acids are the building blocks, amino acids are the building blocks of what? So option number 1 is proteins, option number 2 is lipids, option number 3 is carbohydrates, option number 4 is nucleic acid. So we know that the amino acids are the building blocks of the proteins. So this is a clear-cut question. Question number 9 is, which of the following is a non-essential amino acids? So basically we read what are the essential and what are the non-essential amino acids. So from the options, we can say that the one from the options, tyrosine is the one which is a non-essential amino acid. That is, we do not need to take tyrosine in the form of some supplements or in the form of diet. Simply, it can be synthesized by our body. Therefore, it comes under the category of non-essential amino acids. While if I talk about tryptophan, valine, or lysine, then these are the amino acids which cannot be synthesized by our body, and therefore we need to take these amino acids in the form of the supplements or in the form of diet, and therefore they comes under the category of essential amino acids. So with this, I would like to end today's session. So this was all about the protein. In the next class, we will read about the carbohydrates. This, I would like to end this session, and thank you all for attending it.